Dear viewers, this video is part 6 in the series on cancer, that is Neoplasia, in the Medicine for Common Man series. Coming back to the discussion, early in their development, most human tumors do not induce angiogenesis. Starved of nutrients, these tumors remain small or in situ, possibly for years, until an angiogenic switch terminates this stage of quiescence. The molecular basis of the angiogenic switch involves increased local production of angiogenic factors and or loss of angiogenic inhibitors. The sources of these factors include tumor cells, infiltrating inflammatory cells, example macrophages, or other tumor associated stromal cells, and the extracellular matrix. In the case of tumor cells, several alterations enhance the production of pro-angiogenic factors. Relative lack of oxygen due to hypoxia is one. Driver mutations in certain tumor suppressors and oncogenes favor angiogenesis. Loss of p53 also provides a more permissive environment for angiogenesis. Conversely, mutations in RAS or MYC upregulates the production of VEGF, vascular endothelial growth factor, is a potent angiogenic factor. The idea that angiogenesis is essential for solid tumors to grow to clinically significant sizes provided a powerful impetus for the development of therapeutic agents that block angiogenesis. Bevacizumab, a monoclonal antibody that neutralizes VEGF activity and is approved for treatment of multiple cancers. However, angiogenesis inhibitors have not been nearly as effective as was hoped. They can prolong life, but usually for only a few months and at a very high financial cost. Angiogenesis overview. Vascularization of tumors is essential for their growth and is controlled by the balance between angiogenic and anti-angiogenic factors that are produced by tumor and stromal cells. Hypoxia triggers angiogenesis. Many other factors regulate angiogenesis. For example, p53 induces synthesis of the angiogenesis inhibitor thrombospondin 1, while RAS, MYC, and MAPK signaling all upregulate VEGF expression and stimulate angiogenesis. VEGF inhibitors are used to treat a number of advanced cancers and prolong the clinical course but are not curative. So, dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Invasion and metastasis. Invasion and metastasis are the major causes of cancer-related morbidity and mortality. Local invasion of tumor cells may damage or destroy vital structures and is a prerequisite for distant spread. Although many of these locally invasive cells enter the bloodstream each day, very few produce metastasis. Why is the metastatic process so inefficient? This inefficiency relates to the complexity of the process. For cancer cells to emerge from a primary mass, enter blood vessels or lymphatics, and produce a secondary growth at a distant site, they must go through a series of steps that involve an intricate interplay between tumor cells and many different types of host cells and factors. At each point in this sequence, the breakaway cells must overcome the challenges of 1. Avoiding immune defenses, and 2. Adapting to a microenvironment, example, lymph node, bone marrow, or brain, that's quite different from that of the site of origin of the tumor. The complexity of this series of events may explain why individual metastasis genes have not been found. Mutually helpful genetic and epigenetic alterations that collectively promote the metastatic cascade are required. This set of skills may be present only in rare cells or might even require a collaboration between subclones, with each providing some needed function. Figure 1.8 shows the various steps the cancer cells should undergo to separate from the original cancer mass to travel through bloodstream, and lodge in distant sites. See text. See figure 1.8 metastatic cascade shown earlier. The metastatic cascade is divided into two phases. 1. Invasion of the extracellular matrix, and 2. Vascular dissemination, tissue homing, and colonization. 1. Invasion of extracellular matrix, ECM. The structural organization and function of normal tissues is determined by interaction between cells and the ECM. Tissue compartments are separated from each other by two types of ECM, the basement membrane and interstitial connective tissue, each made up of different combinations of collagens, glycoproteins, and proteoglycans. As shown in figure 1.8, tumor cells interact with the ECM at several stages in the metastatic cascade. To metastasize, carcinoma cells must breach the underlying basement membrane, then traverse the interstitial connective tissue, and ultimately gain access to the circulation by penetrating the vascular basement membrane. This process is repeated in reverse when tumor cells extravasate at a distant site. See the figure 1.8 again. Sequence of events in the invasion of epithelial basement membranes by tumor cells. Tumor cells detach from each other because of reduced adhesiveness and attract inflammatory cells. Proteases secreted from tumor cells and inflammatory cells degrade the basement membrane. Binding of tumor cells to proteolytically generated binding sites and tumor cell migration follow. Invasion of the ECM initiates the metastatic cascade and is an active process that can be resolved into several steps. 1. Loosening up of tumor cell interactions, degradation of ECM, attachment to remodeled ECM components, migration and invasion of tumor cells. 1. Dissociation of cancer cells from one another is often the result of alterations in intercellular adhesion molecules and is the first step in the process of invasion. Normal epithelial cells are tightly glued to each other and to the extracellular matrix by a variety of adhesion molecules known as cadherins. In several epithelial tumors, E-cadherin function is lost due to pathogenic mutations. Note: cadherins named for calcium-dependent adhesion are a type of cell adhesion molecules, CAM, that allows cells to adhere to each other. In many other epithelial cancers, E-adherin expression is silenced through a process called epithelial mesenchymal transition, EMT. It is postulated that EMT is integral to the metastasis of carcinomas, particularly breast and prostate cancers. 2. Degradation of the basement membrane and interstitial connective tissue is the second step in invasion. Tumor cells may accomplish this by secreting proteolytic enzymes or by inducing stromal cells, example fibroblasts and inflammatory cells, to do so. 3. The third step in invasion involves changes in how tumor cells attach to extracellular matrix proteins. In normal epithelial cells, integrins that bind basement membrane laminin and collagens are strictly restricted to the basal aspect of cells. These receptors help to maintain the cells in a resting polarized state. Loss of adhesion in normal cells lead to induction of apoptosis, but free tumor cells are resistant to this form of cell death, termed anoikis, meaning without a home. Additionally, the matrix itself is modified in ways that promotes invasion and metastasis. 4. Locomotion is the final step of invasion, propelling tumor cells through the degraded basement membranes and zones of matrix proteolysis. Migration through the matrix is a multi-step process. This initial phase of metastasis culminates in penetration through the endothelial basement membrane and transmigration into the vascular space. Throughout this phase of the process, tumor cells interact not only with ECM but also with several types of stromal cells, including innate and adaptive immune cells, fibroblasts, and endothelial cells. It is clear that successful invaders induce signals that modify stromal cells in ways that support their malignant behavior. It is easy to imagine that as tumors evolve over time, they come to be dominated by cancer cells that are most effective at co-opting the complex, ever-changing tumor microenvironments to serve their malignant purposes. Vascular dissemination, homing, and colonization. Once in the circulation, tumor cells are vulnerable to destruction by a variety of mechanisms, including mechanical shear stress, apoptosis, and innate and adaptive immune defenses. Nevertheless, viable circulating tumor cells are not rare in patients with solid tumors such as carcinomas. What then separates those few cells that give rise to metastasis from the large number of cells that fail? Also, what determines why metastases ultimately appear where they do in the body? The circulating cells that establish metastasis are much more likely to migrate as multicellular aggregates than as single cells. Clumping of tumor cells in the blood is promoted by interactions between tumor cells and blood elements, particularly platelets, which are believed to enhance tumor cell survival in the circulation. Another potential advantage possessed by tumor cells circulating as a group is that they may be far more likely than any single cell to possess all the properties that are needed to establish a metastasis. Among these essential attributes is the presence of cells with stem cell-like properties, which may contribute not only to the relentless growth of metastatic lesions, but also to the plasticity that is needed for metastatic cells to adapt to growth in a new microenvironment. Where circulating tumor cells arrest and eventually form clinically significant metastatic deposits appears to relate to three factors. 1. Location and vascular drainage of the primary tumor. 2. Tropism of particular kinds of tumor cells for specific tissues. And 3. Escape from tumor dormancy. 1. Location and vascular drainage of the primary tumor. This is a matter of simple anatomy. Thus, colon carcinomas are far more likely to give rise to metastasis in the liver, the first organ downstream of the tumor, than to metastasis elsewhere. However, many exceptions to this rule exist. For example, carcinomas of the prostate and breast preferentially spread to bone. Bronchogenic carcinomas tend to involve the adrenals and the brain, and the neuroblastomas spread to the liver and bones. Such organ tropism may be related to the following mechanisms. Tumor cells may express adhesion molecules whose ligands are found preferentially on the endothelial cells of the target organ. Note, a ligand is a substance that has the ability to bind to and form complexes with other biomolecules in order to perform biological processes. Essentially, it is a molecule that triggers signals and binds to the active site of a protein through intermolecular forces, that is, ionic bonds and hydrogen bonds. Of interest in this regard is the CD44 adhesion molecule, which is expressed on normal T lymphocytes, that's a type of white cell, and is used by these cells to migrate to selective sites in lymphoid tissues. Solid tumors also often express CD44, which appears to enhance their spread to lymph nodes and other metastatic sites. Some cancer cells express chemokine receptors, which may guide tumor cells to tissues expressing chemokines, much in the way chemokines normally act as attractants for cells of the immune system. Even when metastatic cells take root and survive within distant tissues, they may fail to grow. This phenomenon, called tumor dormancy, is well described in melanoma and in breast and prostate cancer. Tumor cells secrete cytokines, growth factors, and ECM molecules that act on the resident stromal cells, which in turn make the metastatic site habitable for the cancer cell. For example, breast cancer cells that are metastatic to bone often secrete parathyroid hormone related protein, PTHRP, which stimulates osteoblasts to make RANK ligand, that is RANKL. RANKL then activates osteoclasts, which degrade the bone matrix and release growth factors embedded within it, like IGF, insulin like growth factor, and TGF beta. These in turn bind to receptors on the cancer cells, activating signaling pathways that support the growth and survival of the cancer cells. Note, TGF beta, that is transforming growth factor beta. TGF beta dysregulation has been implicated in carcinogenesis. In early stages of cancer, TGF beta exhibits tumor suppressive effects by inhibiting cell cycle progression and promoting apoptosis. However, in late stages, TGF beta exerts tumor promoting effects, increasing tumor invasiveness and metastasis. Invasion and metastasis overview. Ability to invade tissues, a hallmark of malignancy, occurs in 4 steps: loosening of cell-to-cell contacts, degradation of extracellular matrix, attachment to novel ECM components, and migration of tumor cells. Cell-to-cell contacts are lost by the inactivation of E-cadherin through a variety of pathways. Basement membrane and interstitial matrix degradation is mediated by proteolytic enzymes secreted by tumor cells and stromal cells. The metastatic site of many tumors can be predicted by the location of the primary tumor. Many tumors arrest in the first capillary bed they encounter, that is the lung and liver most commonly. Some tumors show organ tropism, particularly due to expression of adhesion or chemokine receptors whose ligands are expressed by endothelial cells at the metastatic site. Genes that promote epithelial mesenchymal transition, EMT, may be important metastasis genes in epithelial tumors. Evasion of immune surveillance. Long one of the holy grails of oncology. The promise of therapies that enable the host immune system to recognize and destroy cancer cells is finally coming to fruition, largely due to a clearer understanding of the ways by which cancer cells evade the host response. Assuming the immune system is capable of recognizing and eliminating nascent cancers, it follows that the tumors that grow out in immunocompetent individuals must be composed of cells that are either invisible to the host immune system or that activate mechanisms that suppress host immunity. It is now evident that therapeutic agents that neutralize these mechanisms can lead to tumor regression even in patients with advanced cancers. These encouraging clinical responses constitute strong evidence that evasion of host immunity is indeed a hallmark of many, if not all, human cancers. The following section explores some of the important questions about tumor immunity. What is the nature of tumor antigens? What host effector systems recognize tumor cells? How do tumors evade these host mechanisms? And how can immune reactions against tumors be exploited therapeutically? Tumor antigens. Malignant tumors express various types of molecules that may be recognized by the immune system as foreign antigens. Tumor antigens that elicit an immune response have been demonstrated in some human cancers. Protein antigens that elicit CD8 positive cytotoxic T cell, a type of white blood cell, responses are most relevant for protective anti-tumor immunity. These protein antigens can be classified according to their source and molecular structure as follows. Neoantigens produced from genes bearing passenger and driver mutations. Neoplastic transformation results from driver mutations in cancer genes. These mutated genes encode variant proteins that have never been seen by the immune system and may thus be recognized as non-self. Cancers often have a high burden of passenger mutations throughout their genomes. Some of these passenger mutations, by chance, give rise to protein variants that serve as tumor antigens. Overexpressed or aberrantly expressed normal cellular proteins. Tumor antigens may also be normal cellular proteins that are overexpressed or aberrantly expressed in tumor cells. An example of an overexpressed protein that may be antigenic is tyrosinase, an enzyme involved in melanin biosynthesis that is expressed only in normal melanocytes and melanomas. It may be surprising that the immune system is able to respond to this normal self-antigen. The probable explanation is that tyrosinase is normally produced in such small amounts and in so few healthy cells that it is not recognized by the immune system and fails to induce tolerance. Another group of tumor antigens are proteins that are aberrantly expressed in cancer cells at levels much greater than those seen in normal tissues. This may occur because of gene amplification or because of acquired epigenetic alterations that reactivate genes that are normally silenced in adult tissues. Tumor antigens produced by oncogenic viruses. In several cancers associated with ongoing active or latent viral infections, the responsible viruses encode viral proteins that are recognized as foreign by the immune system. Cytotoxic T lymphocytes, CTLs, a type of white blood cell, play a vital role in surveillance against virus-induced tumors by recognizing viral antigens and killing virus-infected cells. The importance of this immune mechanism is made evident by the high incidence of virally induced cancers that is observed in patients with inherited or acquired T cell immunodeficiency. Many of these tumors are caused by Epstein-Barr virus (EBV) or HPV, that is human papilloma virus, DNA viruses that carry potent viral oncogenes. So, dear readers, this video is part 6 of the series on cancer, that is neoplasia, in the Medicine for Common Man series. Thank you for watching.